The overall goal of this interdisciplinary protocol is to enhance our fundamental understanding of the impact of solvent dynamics on enzyme catalysis.
This method can help answer key questions in the biocatalysis field, such as how solvent reorganization drives enzyme dynamics and catalysis. The main advantage of this technique is that it allows for the engineering of tunnels, or motifs of water transport, which affects specificity, activity, and thermodynamics.
Visual demonstration of this method is critical, as the interdisciplinary steps are difficult to learn because they bridge several subdisciplines of science.
To begin, download a PDB structure of the protein of interest from the Protein Databank. Prepare the structure by removing excess subunits and then adding missing hydrogen atoms and explicit solvent using the cell neutralization and pKa prediction experiment in JSARA. For membrane-bound triterpene cyclases, suitable dimensions of the water box are 91 by 67 by 77 angstroms. Adjust the protonation state of the catalytically active amino acids manually. Minimize the structure by the energy minimization command using the Amber force field suite. Use the particle mesh UALD approach to account for long-range electrostatic interactions. Set the cutoff for van der Waals interactions according to standard settings.